everybody. I am Rachael Samuel, and I work with the Department of Animal Breeding and Genetics, Federal University of Agriculture, Abeokuta, Ogun State, Nigeria. I am also a postdoctoral fellow at the Department of Animal Sciences, Purdue University, West Lafayette in United States. And my area of specialization is livestock genetics and biotechnology. And I have interest in behavioral genetics. And I've been working on welfare and behavior of turkeys and chickens since 2019. And I work on behaviors such as fear, aggression, play, oral-related and comfort behavior in turkeys and chickens. So I want to welcome everybody to Animal Welfare Group Nigeria with the acronym AWGN. So most time we call it AWGN. And the full meaning is Animal Welfare Group Nigeria. This group was founded in 2019 after a gathering of students and lecturers from our College of Veterinary Medicine, College of Animal Science and Livestock Production, and Department of Aquaculture and Fisheries Management, all in the Federal University of Agriculture, Abeokuta, Ogun State, Nigeria. And AWGN has 3 missions. The first one is to increase awareness in animal behavior and welfare in Nigeria, Africa, and the world at large. So that's our first mission. So we want to increase awareness about animal behavior and animal welfare. The second mission is to foster collaboration and networking among researchers who are in the field of animal behavior and welfare. So we want everybody in the field of animal behavior and welfare to work together. People from South America, people from North America, people from Europe, people from the UK, people from Africa. We can do a collaborative research. It's going to be a consolidated one, and it's going to be a robust research. And number 3 is to educate the public on the importance of animal welfare. So we want everybody to know about animal welfare. We want them to know about the 5 freedoms of animal welfare. We want people to know that animals are sentient beings and they should not be maltreated or malhandled. And we meet every 1st and 3rd Wednesdays to discuss issues that are related to animal welfare and behavior. And people from different parts of the world, they have met and we have discussed issues that are related to ethical practices, slaughtering, fear, stress, cognition, neuroscience, vocalization, archery practices in animals like livestock and wildlife animals. And we have 5 scientists that have presented this year. Last year, we have like 19 scientists that presented their work on this group. So this year, we have 5 that are presented already. And today's presentation is going to be the 6th in the series of 2022. And today's presenter is Dr. Rachael Miller of the University of Cambridge and Anglia, Ruskin University in UK. Dr. Rachael will be talking on ManyBirds, a multi-site open science collaborative approach to avian cognition and behavior. And today's moderator is Valentine Obiasogu. And I would love to hand over to the moderator to continue the program. Valentine, over to you.
Thank you very much, Dr. Samuel Durusaro. Hi, everyone. My name is Valentine, and I'll briefly describe myself as an animal welfare enthusiast, almost one welfare enthusiast. Welcome to Animal Welfare Group Nigeria presentation. And as Dr. Durusaro has rightly introduced our presenter earlier, we have Dr. Rachael Miller from the University of Cambridge and Anglia Ruskin University in the UK, and I'll be reading briefly her biography. Dr. Rachael Miller is a comparative psychologist and behavioral ecologist with extensive expertise in animal cognition, behavior, welfare and conservation, and child development. Her research focuses on the evolution of cognition using comparative and ecological and developmental approaches, primarily in birds and children, with a particular academic output and teaching record. Current primary research is in cognition in animal conservation, applying cognition research to avian conservation initiative and the ManyBirds Project, which is a big team open science approach to avian cognition and behavior. With no further ado, Dr. Rachael Miller.
Okay, so thank you so much for that introduction. As you said, I'm Rachael Miller. I consider myself to be a kind of comparative psychologist, but as I'll talk about in a second, I have slightly unusual background in that regard. And I'm really delighted to have the chance to come and have a chat today about our ManyBirds project. And I'm representing our core team of wonderful researchers here. As you said, it's a multi-site open science collaborative approach to avian cognition and research and behavior research. So just really briefly, my kind of background. So very much in research, as I said, working within animal cognition and behavior, primarily in birds and kids. And also quite kind of unusual, I guess, practical experience in terms of hand rearing and training birds. And prior to getting involved in research as an academic, I was actually a zookeeper at RZSS Edinburgh Zoo. So I have practical experience in terms of animal conservation, welfare, husbandry, and training. I also had the pleasure of coming out to Uganda to spend a bit of time with the wild chimps at the Dongo Conservation Field Station many years ago. And I've kind of been all over the place slightly in terms of where I've worked. But as you said, I'm now back in the UK working at Anglia Ruskin University and just taken on a kind of larger teaching position. So very much trying to juggle my research and teaching, as I'm sure lots of people are. Good. So for today's talk, I'm going to focus on just briefly introducing kind of animal cognition and comparative cognition. I'm sure it's very familiar to a lot of you. And also the replication crisis, which has driven the need for open science and also some of the big team science approaches. I'll just very briefly introduce some of the big team science projects and then talk in more detail about the ManyBirds project. And as part of that, I'll introduce you to kind of how we went about setting up the project, what we've done so far. Our first study, which is on Neophobia, which is responses to novelty in birds. And there are lots of opportunities there for collaboration. And also outlining some of our next steps and our plans for where we hope to take ManyBirds. And kind of finishing up with some of the benefits in terms of both fundamental and applied science for animal cognition and behavior. And if we've got some time, I have another couple of slides about that maybe we can go over if there's enough time following questions. We can see how we go. So, comparative cognition, we can define cognition very broadly as including perception, learning, decision-making, and memory. What we try to do with comparative cognition is to derive theories and be able to test these using multi-species comparisons. And this allows us to try to understand the selective pressures that are driving the evolution of cognition. And as you can imagine, this typically requires large and diverse samples. But part of the issue within animal cognition at the moment is that due to methodological constraints and limitations such as funding and the space requirements to hold animals, as well as the labor and the kind of time availability of researchers, we tend to have quite small samples, but within species. And in terms of comparisons between species, this tends to be quite small scale. So for example, just between two different species, which is very, very informative. But it does on a bigger scale kind of limit what we can do in terms of generalizing to across species as a whole. There also have been issues identified in terms of replicability, which I'll talk about a little bit more in a minute. And also limitations in terms of the variation that we can actually include within our research. So of course, there is this ongoing issue in terms of the replication crisis that's affecting psychology and a lot of other fields. driven by aspects such as false positives and the huge publication bias against negative results, small samples leading to low statistical power, and of course, generalization issues that come from this. And as I said, this does also affect animal cognition and behavior to some degree, where we tend to have these less small and less representative samples. And what we really need is kind of multi-site testing and bigger samples so that we can include more species and more individuals within our data sets. So one kind of big response to the replication crisis has been the emerging kind of field of open science. And this means open data, open methodology, source, access, peer review, and educational sources. And some examples of open science practices that we use within ManyBirds and are really becoming kind of common practice, I think, across the field, are things like the pre-registering our data plans before we collect any data, registered reports, making sure that data is being shared openly, codes are being shared openly, open science training, the use of preprints, open access, and repositories so that we can share our data more widely, share our findings, and allow everyone to get involved in what's actually coming out, and the open peer review process to try and reduce some of the issues that happen within the peer review process. And the bigger and wider kind of aims of open science and also big team science are to increase the accessibility, reproducibility, transparency, and inclusivity of research and of science generally. And that this then feeds into the reliability of the work that we're all doing. And this builds also into the 3 R's, so replacement, reduction, and refinement. So this ties into animal welfare and the animal ethics, use of animals in science, and really try to recover some of the lost public trust, I think, that has happened in the last few years in terms of science. We all know that this is a big issue, particularly when it comes to medicine and public health. So big team science is typically encouraging open science, but what we're also doing is building these kind of large scale collaborations so that we can try and address some of these really big questions, such as infant learning and the evolution of cognition. And the idea is that we can combine resources across different sites throughout the world for this common goal. So try to kind of change science and academia from this, big drive for kind of independence and competition to more of a team sport. And I stress that I think we still have to very much have both of these approaches. We need the individual researchers, we need the individual labs, but we also want to be kind of driving some of these big collaborations. And I think it's interesting to note that most of the big team science projects have been founded without any formal funding or infrastructure. So we do face some really big barriers in terms of moving all of this forward. And you can find out more about this in this paper by Coles and colleagues that came out recently. But just to summarize, really one of the biggest issues we find ourselves within ManyBirds as well, is just thinking about how we can reward team players, because it goes against, this academia selfish rulebook, this whole big drive that, you've got to be first author, you've got to be senior author, and how the work is then divided in terms of, how we publish. And as I'll cover a little bit later on as well, big team science and science generally is still really, really not representative across the world in terms of what we should be doing. And so one of the big barriers that we face is to do with diversity, and this can be encouraged and can be overcome in terms of investing the funding and the training and the support required in order to do this. And very much, as I say, most of the projects don't have longer term funding. Some of the really big ones like ManyBabies and the Psychological Science Accelerator do now have funding, and the rest of us are very much kind of setting things up on the side. Certainly with ManyBirds we are. And really on a longer term basis, this isn't really sustainable or scalable. So we really need to be able to attract funding in order to maintain these projects and keep them going. So some of the examples of the big team science projects, this is really exploding at the moment. One of the earliest ones was the Psychological Science Accelerator, which focuses on adult humans, and many babies coming shortly around that time as well. Then there's got many primates, so they've got on about over 150 researchers. And we've got various others like many dogs, many goats has just been launched. I think many fish is on the way. This is really exploding. There clearly is a big drive there for people to be working together and making these projects work, even if the funders are not necessarily quite catching up with it just yet, although I hope we'll soon. And we were really inspired by some of these big projects in terms of creating the ManyBirds Project. And we found, so myself and Megan Lambert, we founded this project in February 2021, and we now have over 50 international collaborators. And our first study, which is open at the moment, is we currently have around 57 species and across 21 countries and counting. So this is still open. Please come and join us if you're interested. I'll give all those details at the end. If you want more information, we have a wonderful website, so check us out at themanybirds.com or our Twitter account. And we do have a few publications now relating to the project, so which I'll cover just briefly in this talk. But you might think about, why birds? Why should we be so interested in birds, for example, in comparison to humans or to our closest relatives, the apes? I mean, one of the biggest advantages of birds is just the sheer size and the diversity of their clade. And there are over 10,000 species. I mean, that's huge. They live across the world. They occupy many different niches. And they really face similar social ecological pressures to mammals and other taxa. And there has been a case suggested for a convergent evolution of intelligence, for example, in corvids and apes. And there's also been lots of really lovely work done looking at the avian brain, such as in terms of the size and the structure and the effects this has on cognitive evolution. There's also lots of really great opportunities for varied research approaches. So from a practical perspective, so for example, we can look at, we can work with birds both in the wild and in captivity, such as at field stations and research stations and zoos. We can look at the effects of domestication. We can look at, we can incorporate citizen science approaches. Birds really are relatively easy to access depending on the paradigms that you want to use. We can make comparisons within species, such as urban versus rural, to have a look at some of these bigger questions to do with adaptability, for example. And so some of the first steps that we took with establishing ManyBirds is that we set up our core team and our infrastructure underlying this, and we outlined this all as transparently as we could within our first article, where we also took the opportunity to kind of launch the project and included a snapshot review. And we're really interested in being able to try to encourage more ManyX and big team science projects and to foster collaborations also between those projects so that we can kind of address these bigger questions, not just within birds, for example, but looking at birds and primates and humans, really making some big comparisons together. And we have a lovely core and study one teams, which are largely early career researcher driven, and at the moment are very Europe heavy. So we're very open to expanding these projects and varying in terms of our collaborative teams as well. And just briefly, we included this snapshot review of avian cognition from 2015 to 2020 across over 550 studies. And we were interested in looking at where are the sites of these research studies. And you can see that it's very heavily coming from labs, 75%. And there's these whole selection of other options that are really largely underutilized, zoos being one of the ones that I'll focus on in a minute. Being a previous zookeeper myself, I think that zoos are just a massively kind of untapped resource essentially for avian cognition and animal cognition research. We also had a look to see where within the world are a lot of these studies coming from, and you can see that there's a massive overrepresentation from certain countries and not from others. And this is very much something that we want to kind of try to help remedy with the ManyBirds framework. And in terms of the species that have been included, there are 10,000 species, but really there is a tiny percentage, less than 1.5% of species that are being worked with. I mean, again, this is in the last 5 years, but just to give you an idea, and really heavily represented by the passerines down here, and typically just across 4 main orders. And we also took the opportunity to make a direct comparison with primates. So the ManyPrimates paper here did a similar kind of snapshot review, and we did it comparably so that we could make these comparisons. And you can see that the sample size ranges are actually pretty similar, and 481 subjects, that's pretty great. But the median samples are considerably smaller, relating to some of those issues that I mentioned earlier to do with methodology and feasibility. And so one of the other first steps that we took was to kind of run a pilot study where we brought together 10 labs internationally to focus on neophobia in corvids. So we had 10 corvid species. This is our members of the crow family, which gave us 241 subjects. And we used for just very simple paradigms looking at responses to novel objects and novel food, which was jelly, and just trying to identify the social ecological drivers of neophobia across these species. And the main thing that I kind of take away from this, as well as it being, just a nice experience and I think quite a nice study, is that there was this really strong desire, again, to collaborate, across labs, pooling resources, let's get together and see what we can do. So I thought that was really, really positive. So just briefly on this study, we were interested in selecting these species because they do differ in their social ecological factors, for example, in terms of their range and urban habitat, etc. So we wanted to explore whether these drivers would have some effect on neophobia. And I'll just very briefly, hopefully show you kind of what this setup looks like, just so you can get an idea of how simple it is. So this is the novel object, novel food condition. We've got our jelly down here. Hopefully this is working for you on your side. And we're just looking at how quickly our birds are coming down to the familiar food. And because you can see that there are quite different, there are some differences in terms of even just to do with the testing environment, and of course to do with the history of these individuals, which is something we have to take into account. We were able to calculate different scores. So we collected data for a control, which is just the familiar food, as well as a novel item condition. And we can then create a different score from those measures. So these are our lovely New Caledonian crows coming down here. So there was quite a variation in different responses to the novel items across different species. And we were interested in how repeatable individuals are, and we found that generally they were quite repeatable when we tested them using the same paradigm across about a 6-week period, and also between different conditions. So that means that one individual who approaches their food quickly in the presence of an object or food, an object tends to do it in terms of when the novel food is there as well. And you can see that we did see that there were some species differences. This is with the novel object. This is the mean latency difference score. And we also identified that there were some social ecological drivers, for example, in terms of the object, urban habitat, adult sociality, flock size, and caching had effect on the responses to the novel object. And in terms of the novel food, just flock size had an effect there. But of course, there are some limitations that we have to be aware of. This was just captive samples. Although we had 10 species, it's still relatively few compared to what's available within the corvid family. And the question remains whether these are corvid general patterns or whether these are appropriate or applicable across birds more widely. So we decided that neophobia would be a great paradigm and focus for our first ManyBirds study. And as I say, we do have an open call now until January next year for data collection on this. And neophobia it's important because it allows animals to learn about and exploit their environments so they can navigate risk and the impacts on their adaptability and ultimately their survival. And we selected this paradigm again because it is so simple. It's low cost, it's low time, it really can be done in any setting, lab, zoo, field, private, and with any species. And we're interested in asking these research questions to do with, what are the social ecological drivers of neophobia across birds more widely? What species differences can we find in terms both within and between? Are there differences, for example, between closely related species that, chickens, for example, that are domesticated, red, jungophile, their wild counterparts? And also how repeatable are individuals? Are they consistent in how they respond? And our next plans for this will be to try to build on, bringing other sites in, for example, zoos. And as part of this, we've kind of identified some of the challenges with working within zoos, both from an animal perspective and also the kind of human, the staff perspective, and try to provide some solutions to this. If you're interested in this, you can check out this paper. And we've come up with some kind of suggestions for designs that are most applicable within the zoo environment, such as kind of drop and go apparatuses, designs that don't require training or close human contact or animal separation. And we're really interested also in developing automated technology paradigms. What we want is something that you can just place it into enclosure, collect data, and that's you done. Because we have to recognize that to test in lots of different sites, you need something that can try to make this as comparable as possible. And our other next steps will be trying to expand on our citizen science approach. We really want to have it so that people can essentially, test their pets, test the birds that come to their gardens, make this really available to lots of different people. And for our next study ideas, we're planning to take a consensus-based approach so that our collaborators can input on both the study focus as well as the methodology that we select for the next studies. And some of the areas that we're interested in, and I think will most likely be able to be worked to be comparable across species, are aspects like self-control and memory, play and exploration and innovation or problem solving. And again, just trying to look at both the kind of taxa and species level as well as the individual level as much as we can. But it does have this caveat that we want to ideally have paradigms that are low cost, low time, low labor intensive, so that we can test across different sites and really kind of make this as inclusive as possible. And we've also identified some areas where we can try to collaborate between different big team science projects. And some of the ones that might be of particular interest to the group today are relating to things like surveys and welfare and enrichment, conservation, broader effects of captivity and also domestication, human-animal relations. And we're in discussion with some of the other big team projects, and it's one of the focuses of the Big Team Science Conference, which is being organized by Many Babies and Many Primates, which is in October. So if you're interested in that, do check it out and come along. And so just to kind of finish up, I just wanted to highlight some of the benefits of animal cognition and behavior research in terms of both fundamental knowledge to do with biology and evolution, but also the applications that are possible to do with conservation and welfare, cognitive enrichment or enrichment of animals that are in captivity, the engagement of the public, in terms of our citizen science approach and just trying to bring people into more closely involved with what we're doing. And as part of this, trying to increase the diversity of this research. And I just wanted to finish with an example, which I've been using a lot recently, but I really like, as an example of, the applications of behavior, animal behavior work, is that, recent research has led to changes in animal welfare legislation. And one big example, I think, being from the UK, is that the UK recently extended the scope of their animal welfare sentience bill to recognise cephalopod, mollusks, and decapod crustaceans as being sentient beings. And this was based on this extensive review of this, empirical scientific evidence of sentience within these inverts. And I think this is just a really, really nice example of, what we're doing can have these bigger, real world applications. And this is very much something that I hope we can do through the ManyBirds Project as well. So just want to say a big thank you to our collaborators, and thank you very much for listening and for inviting me along. Happy to take any questions. And also, if you would like more information, and particularly if you're interested in registering your interest or signing up to collaborate, just head to our website and you can do it all from there. All right, thank you very much.
Thank you very much for your presentation, Dr. Miller. So the question is coming from Dr. Shilni Asere, and she's asking, how did you balance for the variation in different labs when conducting ManyBirds project?
Yeah, this is a great question. And so this is something that we need to try to do with, with the first study, the Neophobia study, we try to balance for this by having a control condition of just how they normally approach their familiar food. And we can then use this to create a different score based on how they then approach when they have this novel item presence. So at least we can somewhat standardize within an individual and within a site as to how they're responding to novelty. And I think something else that we can try to do with this, and we will be doing, is to collect as much information as we can about the subjects and the sites, so that we can include some of these variations within the analysis that we run. So, this can be bigger questions to do with whether they're captive or wild, or the kind of different categories within the site location. But it can also be things like, the type of rearing that they've had, what prior training experience, these kind of things that we can incorporate within the analysis. It's an issue for big team science projects generally, but I think this is also very much an issue for any kind of any other animal cognition or behavior research. Essentially, is that you are going to have, uncontrollable aspects to do with the site and to do with the history of your subjects. But the more that we can include this within analysis, the more we can identify how much these differences might actually be driving, cognitive performance, for example.
Thank you very much. And the next is coming from Said Sabet. He says, great and an interesting presentation. I was wondering how multi-site open science collaboration approach may initiate and facilitate potential multidisciplinary research and scientific collaborations.
This is very much one of the aims of the big team science projects, is to be able to bring people together and pool our resources. And the ways that we can try and to do this, I think, is really through this kind of framework that we've just we've talked about. Try to come up with paradigms that can be run as comparably as possible across different sites, and open up for collaboration, so that people who are interested really can just sign up. Come and join us. Follow this very clear protocol that we've got. Within some kind of caveats to do with authorship and these kind of guidelines that we have in place. But this is very much something that we want to make happen. And I think if you look at some of the more established projects, such as ManyBabies, such as ManyPrimates, you can see that they have really they seem to be really successful in that sense. They have authorship pages and pages of 200 odd authors, which must be a nightmare to put together for the publication. But I think those are great examples to show that there really is this drive, and that you can be quite successful in doing this.
And just a very quick one, I want to ask, can we say that there's a similarity between the multi-site project and iNaturalists? If you have heard about iNaturalist.org before, where they collect data on roadkills in different parts of the world, because I was wondering how you were talking about people in different parts of the world participating in this research. How will they be able to remotely gather this data and then collate the data for the research? Is it something like that kind of framework, or there's a different approach to it?
Yeah, I mean, so I don't know the one that you mentioned, but I'll definitely check that out. I mean, so all of our, through our website, you can find all of our sign up documents. And so everything is on there to do with the protocol, to do with how you collect the data, you code the data, you input it into the data sheet. It's like everything is on there, so it can all be done remotely. I mean, even our core team are not in the same place. I think I'm the only one in the UK at the moment. The idea is that you can follow the guidelines that we have there. You are responsible then for coding and inputting your data, making sure, for example, that there was inter-observer reliability done on the data during the coding to make sure that it's being coded reliably, following the guidelines for how we put this into the data set. And then we'll have an analysis team that then deals with this. And how you collect the data essentially depends on which species and where you're planning to collect it. I mean, if you, for example, you already have a lab that you work with, you could collect the data there. If you already have a field site you work with, you have a local zoo, you can set up your own kind of collaboration to have access to animals to be able to then collect data for that.
I think it's pretty similar to iNaturalist. And this is coming from Dr. Samuel Durosaro. He says, how reliable are results from preprints, bearing in mind that they have not gone through editorial and peer review?
Yeah, it's a great question. And I think it depends a lot on the study, and we have to be very careful about interpreting preprint data and preprint interpretation of data. And I think that there are pros and cons with preprints, and certainly for aspects, for example, to do with like public health, we've seen a lot of this to do with COVID. We have to take a lot of care that these preprints are not, going to the media, are not being fed into legislation changes. There's not big actions that are coming just from the preprint. We shouldn't be doing that. We should be, waiting until it's gone through peer review before you're making those bigger changes with that. But some of the big benefits of preprint, is just being able to really disseminate our research, what we're working on, open things up so that discussion can happen during the peer review process. As we know, the peer review process itself is not without issue, and just because a paper or a publication has been peer reviewed also doesn't necessarily mean that it's reliable data in itself. I think there's a lot of examples of this. So in terms of how reliable they are, I would say treat it with caution, but I think there are a lot of advantages to using preprints as long as there's this strong caveat that this should not be being used more widely. The media should not be jumping on it and spinning the story as they tend to do until it's gone through peer review. But it really speeds the process up. I mean, it's particularly for early career researchers. I think it's been an absolute, it's been such an important change to be able to get our research out there when we need it, not 3 years later.
Thank you, Dr. Miller. This is coming from Site 7, and he's asking, is feather plucking a behavioral disorder only in captive birds? And can it be seen in wild birds as well? And does it have a relation to neophobia?
Yeah, it's a good question. I don't know actually whether feather plucking is also seen in wild birds. I can't think off the top of my head of great examples. We know it's a big problem that we find in a lot of captive species. I know that some zoos, for example, even really, really great zoos deal with this with some species. And there's been a lot of work looking at this kind of stereotypical behavior and what we can do to try to remedy this in terms of behaviors and enrichment, and whether it relates to neophobia. I mean, it's a good question as well. I don't know if we know for sure. It could be if we're thinking about neophobia in terms of a kind of stress or fear response to novelty, then potentially that could drive a kind of stereotypical response like feather plucking. But to my knowledge, I don't think at the moment we know for sure that those are connected, but I think that it definitely needs future work.
Thank you very much, Dr. Miller. This coming from Dr. Samuel Jerusalem is asking, what are the constraints you are facing in ManyBirds research?
Yeah, I mean, the biggest constraints I find personally is that, as I kind of touched upon, without formal funding behind the project at the moment, it's almost sort of done as a side project alongside all the other things that we're all trying to do. And to some extent that works okay because of the nature of, you know, having a team behind the project, having lots of collaborators involved in the project. But there's still quite a lot of work, as you can imagine, to do with, you know, project management and kind of making some of these the bigger decisions and just keeping it all kind of moving along. So very much what I would like to do is try to attract some funding, and some of the other big team science projects have been successful with this. You know, they now have dedicated staff on the project, and I think that would help a lot. And I've also been collecting my own data to contribute to, you know, to the first study. So you know, you do have to think about just the time and kind of energy and investment in that as well. So yeah, I think just those are the most practical constraints I can think of that come to my mind.
Thanks. This comes from Dr. Shioni Yasser, and she's asking, what are the implications, or what is the implication of the increase in heat wave, global warming on animal cognition?
Yeah, it's a really big concern, isn't it? I mean, on the animal or on the research, I mean, if we're thinking about the animals, yeah, I mean, it's going to be scary. I mean, we were talking at the beginning of this meeting just about, you know, the wildfires, for example, that, you know, have been flooding the UK and Europe just in the last couple of days with this heat wave. And you know, you really worry about how this is impacting on our wild animals, and what we can be trying to do, even just in the short term, to try and support wild animals, you know, whether that is having to try and give them access to water to drink and to bathe, or to extra food. And I think, you know, there's some of these much bigger questions to do with the long-term changes that we need to make as individuals, but really also our governments need to make in terms of what we can do to remedy the issues to do with climate change and global warming. And yeah, I mean, I guess just in terms of how this will affect research, one of the things that comes to my mind is again going to be about funding availability. I mean, I think funding, I don't know how it is in the rest of the world, but certainly in the UK, my feeling is that funding is very much moving away from kind of basic science to applied science. And there is just less money available to do kind of basic animal cognition and behavior research, for better or worse, you know. So if we can start thinking about whether there are smaller changes that we can make for some of these animals based on some of our findings, then we're also hopefully more likely to attract funding so we can actually continue this research at all.
Thank you. This is coming from Dr. Samuel Jerusalem, and is asking, what is your perception about authorship? Who should be the first author in a collaborative research or big team science?
Yeah, so the different projects have taken slightly different approaches with this. So I don't think that there's kind of one strict answer. And you know, for example, so many primates actually use many primates as their first author, and they will then use, don't quote me on it, but I think, you know, alphabetical or some kind of random assortment for the other authors. And what I don't think we've made a formal decision on this yet. We're tending towards the idea of using ManyBirds as the first author to kind of overcome some of these issues that come with authorship orders. what we did with the Corvid paper was that we kind of assigned authorship to the sort of leadership team. And then everyone else, the rest of the collaborators, I think, were alphabetical within that. So there were lots of different approaches with that. I think many babies, off the top of my head, tend to go for the more sort of traditional kind of first author, being the person that, you know, has most strongly led the project, may have written the paper. For me, I think for ManyBirds, it's going to depend a lot on what actually happens with the first study. Because what we would really like is that we can kind of get quite a few people involved in all aspects of bringing together any publications from this. So it's not the traditional kind of, you know, collecting data and then a few people do the writing of the paper, the analysis. What we'd really like is to kind of have small teams responsible for each part, for writing the paper. This team's responsible for the analysis. And so that there's less issue to do with kind of how you divide the authorship stuff following that. But yeah, it's an important consideration, definitely.
And thank you. And Dr. Cheon Yasser is asking, do you consider the age, sex, degree of domestication of the birds used in the project?
Yeah, definitely. So we'll be collecting, so on it within our sign up documents, we ask for basically all the history that you can give us, or the information that we can get on our subjects, so that we can look at age and sex and domestication and other kind of aspects that are, you know, going to differ between the individuals and between the different sites. So we're also asking about site kind of details with that. And again, as I said, so that we can try to control for some of this within the analysis that we run. And definitely I would be interested in age, because there's a lot of research showing that there are quite a lot of differences in neophobia between juveniles and adults. It can go one way or the other depending on the species. Typically juveniles tend to be less neophobic than adults, but this isn't the case for all species. Hawaiian crow jumps to mind as one that has the opposite pattern. So I'd be really interested to see kind of how it, you know, what the outcomes are of that, and we can make predictions in both directions.
And she's going further to ask if you have done any neophobia research in chickens.
I haven't personally, but I would love to include chickens in our sample. So please come and join us. I would be really interested in it, and particularly if we can also have a sample of junglefowl, for example, so that we can kind of see, you know, start sort of teasing apart what the effects of domestication might be on neophobia. From the top of my head, I think that, again, depends on species, but domesticated species tend to be less neophobic, I guess, particularly if they're raised closely to humans. You know, they're much more used to these, you know, changes into their environment than their wild counterparts. So yeah, please come and join us. Test some chickens.
And then Dr. Samuel Durusari is asking, for the state of the avian cognition field, you classify the study sites as lab, field, zoo, farm, and others. What are the components of these others, in quotes?
I think that this would have been like private. So, I mean, I think that was a tiny component, but I think it would be, you know, things like private establishments. So pets, I think probably would jump in there. I don't think that anything within that review that we did actually included citizen science approaches, off the top of my head with that. But as I say, I mean, I think that just, you know, that kind of simple presentation really highlights how much of the research is coming from the labs as opposed to some of these other sites, which is such a shame, because in particular, you know, the zoos hold however many different species, you know, often threatened species, And the more information that, behavioral information that we can gather about some of these threatened species, the more that we can consider whether we can actually apply some of these findings back into conservation, for example. And this is something that I really am, this is one of my other projects that I'm really keen on and trying to work on is with ballymina, which are critically endangered species. And so trying to kind of work with them within zoos where we can access them to learn about their cognition and learn about their behavior, in particularly things like behavioral flexibility, things that might be relevant when you're, when they're actually getting reintroduced, which they are part of active reintroduction programs, to see whether we can improve that process, to do with pre-release training and other changes like that. Yeah.
Thank you very much, Dr. Miller. I was wondering if there was any further questions and anyone wanting to ask any further question, you can unmute yourself to ask.
Thank you so much, Rachel, for the wonderful presentation. And it's good to know about your work on ManyBirds. And I'm so glad to have you present today. At least I now know that there is a ManyBird project, and which is open to all kinds of bird species, including, the jungle fowl, the domesticated chicken, which is the area I am into. I work with the Nigerian indigenous chicken, but at the moment I'm working with 2 German breeds. I'm working with the Silkies and the bushfowl. The German bushfowl at the moment is almost close to the red jungle fowl. That is the closest they have to the ancestral chicken sort of. So I'll be happy to collaborate with you on this project, because in your presentation I took some slides, the screenshot, and I saw that from the map, the demographic stuff, little has been, contribution has come from Africa, even as a continent, in terms of avian cognition. Though actually, I think I had a publication sometimes last year on chicken cognition using one of the improved native chicken in Nigeria, which is the Funab Alpha chicken. I think it's one of the few that we have, really. So with that, I want to say you are doing a good job for bringing researchers from all over the world to join in a project. And thank you for being open to collaboration. Thank you so much.
Oh, fantastic. Yeah, thank you so much.
So thank you very much, everybody. And I am so grateful, Dr. Rachael Miller. I got so many new things today. And I was so surprised that I didn't see any research work from Africa. So you will need to explore Africa. And we have so many people that work on cognition in Africa also. I think I've done some work on turkey cognition, though we have not published it. Then Dr. Yasser has also done some work on cognition. So we are all open to collaboration, and we would love to join the project. And we also love to thank the foreigners in our midst. Sahid, thank you so much. Sahid has been part of our program since the day he knew about this program. Sahid.
Everyone, thank you very much for a great presentation, Rachael, and thank you, Elevation, for organizing such an interesting event. So I really enjoyed the presentation, and very interesting and great follow-up discussion. So I'm really, great that contributed in this webinar. I'm looking forward for next presentations, of course.
Thank you very much, Said. Said is working on fish vocalization, bioacoustics, and fish welfare. So if you want to learn anything about fish welfare, you can always contact Said. We also have some other foreigners in our midst. We have Jao Polo.
No, I just want to say thank you for this presentation. It was very interesting to see all the projects that you have over there, and to the Animal Welfare Group of Nigeria. I want to thank, because all the presentations that you have been organizing have been really cool, and it's a great example for all the research. And yeah, it's cool to know what is being done in other parts of the world. That's it.
So
Thanks.
thank you, Sony. Thank you so much. Sony has been joining us. I think I can see some other people. Thank everybody for participating in today's presentation. And I would love to appreciate Dr. Miller. You did a wonderful job, and I pray you get funding for this wonderful work that you are doing, because you will need a lot of money. Sometimes you will need to travel down to see what other people are doing, see how they collect their data. So my prayer is that you are going to get funding and you are going to get a lot of money, because it's a good team. You have a good project that you are doing, and I would love you to cover all the continents in the world, especially Africa. Don't mind me, I'm a little bit biased about Africa because I'm from Africa. So we are so grateful to have everybody in our meeting today, and we hope to see you in our subsequent meetings. So you can join us on our Facebook and LinkedIn. Our Facebook and LinkedIn, we are on Animal Welfare Group Nigeria. So Animal Welfare Group Nigeria, if you want to join us on Facebook, if you want to join us on LinkedIn. And our Twitter handle is at AWGM14. AWGN14, 14 in number. 14 in number. Then the next presentation will be on the 5th of August. And it's going to, the 5th inaugural lecture, rather, sorry. The 5th inaugural lecture will be delivered by Professor Lisa Young on August 3rd. August 3rd, not 5. August 3rd, 2022. And she will be talking on zoo and wildlife welfare. So for people in zoo, it's your turn to talk. So come and listen to Professor Lisa. And Professor Lisa is from University of Nottingham in the UK. So if you want to learn about wildlife and zoo welfare, it's a great opportunity. So Professor Lisa will be talking for almost 90 minutes on zoo welfare. I'll be there, and I would love to learn a lot of new things about zoo and wildlife welfare. And I know Said will also be there. So the 55th Congress of the ISA, that's International Society for Applied Ethology, will be holding from September 4 to September 9, 2022 in North Macedonia. I would love to enjoy our people to join that meeting physically. We are going to meet a lot of people, and we are going to have chance to collaborate with other scientists from other parts of the world. So to everybody that participated, I'm so grateful. I say thank you. And to Dr. Rachael Miller, you are doing a very great job. I love your presentation. And please make sure all the continents are covered. And make sure you cover as many birds as possible: chicken, turkeys, ducks, and so many other birds. And we are going to be having a program in which myself, I'm Rachael Samuel, Dr. Yasere and Vito Yenio will be presenting. We'll be talking about maternal behavior in 2 ecotypes of chicken. So watch out for that event. We are going to paste it on the LinkedIn, on our Twitter handle. It's going to be informative and educative. So Saeed and other foreigners, thank you so much. And to the Nigerians, thank you for always joining us. I'm so happy that we do this together. Valentine, thank you for moderating. Dr. Oluwaseun Yasere, thank you so much. We really appreciate you. Dr. Majek, Dr. Bukola Majakudumi, thank you so much. Jatutu, thank you so much. Olumide, Barnabas, Oluwatosin, Zeno, Promise, Victor, Young Ilioji, Dr. Young, thank you so much. Afusat Lade Jobi, Dr. Komolafe Priscilla, and every other person that joined us today, we say thank you. And we hope to see you in our subsequent meeting. Have a good day. Thank you so much.